Welcome everybody to the Kennisclip Dentinogenesis. My name is Sarah Pringle. I'm going to be reading you through this Kennisclip. Let's get started. You probably know this will probably look familiar to you already, but the hard tissues that we need to understand how they are formed, that you need to understand how they are formed, are these three tissues here. So there's this left hand tissue which has kind of these waves patterns in it, can also look like this if you turn it the other way around. You have this middle tissue which has what look like holes in it, and also these patterns of cells, and this right hand tissue which looks a little bit like a tree if you look at it in a particular angle. So probably these things look familiar to you. So the left hand side is glasur, enamel, dentine or cement. Today we're going to look at how you make dentine, which cells are responsible in more detail. We started looking at it in the process of embryogenesis of the tooth, and now we'll go more into specifically the life cycle of the cells that make dentine. So a quick recap, this should hopefully look quite familiar to you. What is dentine? Dentine is, of course, here the blue stripy structure which makes up the bulk of the tooth of the body. So it's a huge portion of the tooth, and it's really important that we understand how it is made. It's harder than bone, but it's softer than the enamel on the outside. It's made of a lot of closely packed tubules, of which dentine is 70% hydroxyapatite and then 20% organic components, which you will learn more about in future lectures. It is also kind of bound, stuck to the enamel at what is called the dentino-enamel junction. So that would be around here. Which cells make dentine? I hope maybe if you watched recently the Kennisclip that I recorded about embryogenesis, that this will look a little bit familiar to you. This is the bell stage, the clock stage, the late bell stage, and what you can see by this point of tooth development is quite a complicated looking structure. So you have, is the dental pulp, sometimes called the dental papilla. These are the odontoblast cells which have differentiated from the dental papilla. These are the cells which will make the precursors of dentine, which is in yellow here in this cartoon. On the outside, or here, you have the ameloblasts, which will make the enamel, or the precursors for enamel. And these cells are derived from the inner enamel epithelium, if you remember this layer here, the outside being the outer enamel epithelium, this being the stellate reticulum, and this is stratum intermedium. So for the purpose of this Kennisclip, we're going to focus on these odontoblasts here and how they make the precursor forms of dentine. So dentine in yellow, odontoblast labeled here. Dentine is made by odontoblasts, starting at this last stage of bell stage development, and I'll take you through these stages slowly. So here you have a cartoon version of, let's say we zoom in on this part right here of the developing tooth and we enlarge it. This is a dental papilla cell. So at this moment it's not making any dentine components. The epithelial cells on the other side, I hope you will be able to understand as being the inner enamel epithelium, which will turn into cells capable of making the precursors of enamel. So we need to get these cells turned into a type of cell that can make dentine. And in order to do this, the cells here receive a signal from the epithelial cells, actually, from the inner enamel epithelium. So already you can see there's a crosstalk between the two cell types. This signal stimulates a change it stimulates the division, proliferation of these cells. So this cell here is one cell, and this cell is now, I hope you can see, dividing into two cells. So you have two future nuclei developing. When these cells are divided, you will see a change in morphology. So there are two cells now. Imagine these have come from here. This cell looks very much similar to the first one that we had over here. And that's a very logical conclusion, because this is a sort of cell that is kind of saved for the future. It's a reserve type of cell in case we need to make more dentin. These cells are now called pre-odontoblasts. You can see they are developing into cells with a longer and bigger cell volume than the dental papilla cells with more cytoplasm. Logically, this is useful because the cells need to be producing a lot of proteins that need to be secreted from the cells. So that's step 3. So far you have the dental papilla cells, you have their division, you have their differentiation into pre-odontoblasts. And then you have their formation, their full differentiation into odontoblasts producing the proteins that make up dentin. So be careful with dentin being everywhere because actually it's pre-dentin that is made by odontoblasts, which is then eventually mineralized into dentin itself. Because dentin is a very hard tissue, and if a cell was to try and produce fully formed dentin itself, it would die in the process of secreting it. Noticeably, this is a really important difference between this cell here to this cell, so you need to consider them as this is the early stage and this is the later stage. This odontoblast now, these odontoblasts, this is the mature form, have a really long cell body with cytoplasmic extensions, they're called, so all these kind of branching, tree-like structures here. And if you were to look back into the microscope, this is what you see reflected in how dentin looks. So all of these holes are where the cell bodies, where these cytoplasmic extensions would have been. Around the outside, the grey is the dentin itself, and the holes would be where the cells were. So you have a transition from dental papilla cells and a sort of maturation into cells capable of producing pre-dentin. How does this look under the microscope? This is important for you to understand. You will be expected to know this and to be able to identify odontoblasts microscopically. So again, focusing on this last stage of tooth embryogenesis, if you zoom in on this area here, so on the inside here we have the dental pulp, dental papilla. Here you have, this would be the striatum intermedium, the stellate reticulum. This would be on the outside the ameloblasts. We won't focus too much on them now. But here, if you look at this transition from the kind of square, smaller cells that we were talking about, if you move with your eye up, you sort of start to see these longer cells, so OD, odontoblasts. And using this particular staining which they've used here, blue in the middle is the dentin which is produced, the pre-dentin. And if we zoom in again, you can now see exactly what I mean with the shapes of the cells. So this, I hope, is very obvious for you to see is the nucleus of an odontoblast, and this is all of the rest of the cell body, which would actually go all the way down into here, into these holes, these canals. So from here to the right, this is the dentin. And what I hope you can see is that this lighter pink stage is the pre-dentin, which is what the odontoblasts secrete. And this darker, kind of dark purple stage is the mineralized final version of dentin. So this has been processed and is now a hard tissue. Really important to understand, and I think I've mentioned it before, that dentin formation happens before enamel formation. Even though the cells in the dental papilla get a signal from the cells in the inner enamel epithelium to begin their differentiation into odontoblasts, the enamel epithelium does not start its own transformation to make ameloblasts until the dentin production has happened first. So you see here, dentin, odontoblasts, ameloblasts, but no enamel yet. So in summary, this is quite a short Kennisclip because what I will focus on in the next clip will be also bringing together the process of dentinogenesis and amelogenesis. Is this last stage of embryogenic tooth formation and these 4 phases of the odontoblast's basic life cycle. So the dental papilla cell, its proliferation, the transformation of one of these daughter cells, is what you can call it, into a fully functional odontoblast with these long cytoplasmic extensions, which are producing the precursors of dentine. Here you can see them, I didn't mention it, but these small blue dots are supposed to represent the precursors of dentine produced by the odontoblasts. And microscopically, it's quite easy to see where they are, and also even easy to see the mature and immature forms of the dentine. So next time, to start to bring this together in the next Kennisclip, I will talk about amelogenesis, and then we need to go into a more deeper consideration of the interaction between the odontoblasts and the ameloblasts, because they, I hope you're beginning to realize, are very dependent on each other for the successful formation of teeth. Information sources are these guys here. I understand that you may or may not have the Nancy book on the left, the book on the right, the Berkowitz you should have, and also the same tips to look at some videos online, but look critically. Okay, thanks for watching, and until the next Kennisclip.